Book review Beyond Inheritance. Eris Topol in discussion with Roxanne Kamsi on her phenomenal book Beyond Inheritance. It analyzes the critical impact of our acquired, somatic mutations impacting our health and disease. Tagline: we are all genetic mosaics.
We have really a phenomenal topic and new book that was just published today by one of the leading science journalists there is in the North American and the world, Roxanne Kamsi. She is an Atlantic journalist for science, and she has come up with a mind-bending book called Beyond Inheritance: Our Ever Mutating Cells and a New Understanding of Health. And I think you'll find a new understanding. This is one of the most, I think, biologically provocative books I've read in some time. I'm really excited. I've known Roxanne for many years, and it's such a delight to welcome her to Ground Truth. So hello there, Roxanne.
Thank you, Eric. Thank you for having me. I have known you for a while. I've been a super fan of yours for a while. And I appreciate, I think there's some overlap between what I wrote about in my book and what you've written and talked about. And so I'm excited to talk about it today. It's going to be great.
Oh, it's so much there. I mean, basically, we're all these genetic mosaics, and you have to wonder, with all these mutations that are occurring in the body, why aren't we just falling apart? So I just wanted to frame this with the math. And by the way, so many of you are joining, and if you have questions that you want me to get into with Roxanne, just put them in the message, and I'll try to as we forge ahead. So the math is striking. We have somewhere around 37 trillion cells. Of those, every second, 4 million are turning over, or 330 billion per day, trillions of mutations per day, and 2.4 million new red blood cells per second. So we have lots of opportunities for these mutations to take place. I mean, no shortage of them.
Absolutely. Yeah. And what strikes me is that, this all begins with one cell. So you, me, everybody listening, we were all, for the first 24 hours after we were conceived, we were just one cell. And then 24 hours later, we became 2 cells. And then the math goes from there. Like you say, by day 5, we're 100 cells. We've got a hollow center. And every time that division happens, there can be errors that occur in the copying of the DNA from one cell to the daughter cells. And those mutations then get kind of instilled to the subsequent cells. So you're kind of accumulating mutations throughout life. And when you start from one cell and we end up with this multicellular existence, things can get complicated. And I think there's some really medically relevant mutations that happen at different stages of our lives that we haven't been paying attention to. So I wrote this book to draw attention to this kind of massive shift that's happened in genetics that a lot of the public just hasn't been told about. And I felt like I had to tell somebody, I had to write about it.
Well, you sure did. And a lot of people conceive of mutations as really bad. And that means that you're going to have cancer or die. And you bring out the other side of this as well, which we'll get into. So the key point as a foundation is that with all these cells in our body, they're constantly in a state of, as you call it, endoevolution, or a Darwinian selection of our cells. They're competing with one another. And one of the things that really highlights, and we're going to talk about 3 papers in the last week, including one just today, that really highlight the work that you've reviewed and all your insights in this new book. But in recent years, certainly in the last decade, we've seen a lot of single-cell sequencing of DNA, RNA, the nucleus. And has that been a big help in understanding this whole mutation story?
I think absolutely. So a large part of what brought me to this topic was the fact that single cell sequencing, so the idea that you could individually sequence cells, not just bulk tissue, which is important in of itself, but you can find out a lot about the proportions of cells in a tissue that have a mutation. And this was really important in one of the cases that I talk about in my book. So there was a family in California in 2013 that had a daughter. And when she was born, doctors saw that she had, and you know where I'm going with this because it's a cardiac story, but she had what looked like to be long QT syndrome, which is where there's an irregularity in the heartbeat. And they analyzed her cells, and with the single cell sequencing technology, they saw that 3 out of 36 of her blood cells had the mutation. So they tested her parents, but neither of them had this long QT mutation, which was shocking because this was thought to be an inherited disorder. So through a series of steps, they actually confirmed that this had spontaneously occurred in her development. So when she was at that embryonic stage, a long QT mutation had spontaneously occurred and gone on to form her heart tissue. And I think this is really important because it shows how this sequencing technology is changing our thinking about diagnostics. So whereas we might have said, oh, this is only an inherited disorder, we can now stop and say, okay, maybe we should consider that this disease spontaneously occurred in somebody.
Yeah, I mean, this is so important because so many of the insights that you go over and help explain some very complex stuff is new because we have these tools to unwrap.
Yes.
And as you say, what we call a phenocopy, which is
Right.
here, this young girl had a long QT, which of course can be inherited, but hers was not inherited. It was a spontaneous mutation. So it's a copy of what, would occur with a germline mutation, here a somatic mutation. Now, this was striking for me in that although you're writing this and publishing this book today in 2026, this guy Wilhelm Roux in
Yes.
1881
Yes.
wrote a book before any of this stuff, Struggle of the Parts.
Yes, yes. Der Teil in there's a whole German title that I have usually an ability to say, but it's long. And he basically read Darwin's book. What's fascinating is that he wasn't meant to be a scientist. So he came from a family of fencing experts. They had hoped that he would become a fencing expert, but he was fascinated by biology. And he grabbed Darwin's book when he was young, and he just loved it. He thought this guy's onto something. Of course he was. And what this Wilhelm Roux character did is that he applied Darwin's thinking to the body, and he said, you know what? We know about cells. What if cells compete against one another the same way that species compete against one another? Or what if natural selection is happening in the body? And he actually got a copy to Darwin, and Darwin said, this seems really interesting. It's in this German I can't totally understand. But what you mentioned is totally true. This guy, Wilhelm Roux, kind of foresaw all the things that we're talking about today. He just didn't have the technology to be able to confirm it.
It's amazing. And then the fellow Nietzsche that wrote after uniformity of is
Yes.
pure delirium inspired.
Yes. Love that quote.
This
Yes. Nietzsche wrote that.
uh,
Yeah.
this I didn't even know about Struggle of the Parts. I mean, I
I
just
didn't either until I started this, and
ah,
I'll tell you, it just got translated into English in 2024. So
oh,
and I've been working on this book for 3 years. So at the first in the first year of working on this book, actually first year and a half, I was just kind of like struggling in my like old German knowledge to figure out what he was saying, and that just shows you though how people are coming back to this idea, right? The fact that not only was I interested in this and uncovering this, but also somebody decided to translate his book.
yeah. Well, a good thing they did. And what prescient book? They got
Yeah,
1880.
yeah.
So I mean, this is amazing. So go ahead.
And I was just going to say the other thing that happens to be a coincidence is that Ernest Teisler, who's at Harvard University and is a doctor, he actually 100 years ago this year, so in 1916, he used the phrase somatic mutation for the first time. So we're actually at an anniversary of this understanding of somatic mutation, which is a term to describe the mutations that happen in the soma, so the body, as opposed to our reproductive cells.
Right, right, exactly. And we'll get into some of the easiest ones for people to understand, the ultraviolet light, the skin
Yes.
mutation,
Yeah.
and we'll get into that. But before we do, one of the most important areas that you lay out a whole new path is in cancer. And again, we have the cellular competition with winners and losers. And right now, there's a transition, but for most of the time when we had cancer, it was carpet bombing, kill every cancer cell. Now,
Yes.
that will be the wrong idea, as you've reviewed, and the whole idea that maybe that's not adaptive cancer treatment, leaving some cancer cells. Can you explain why that has seemed to work, at least in prostate cancer and perhaps in other types of cancer?
Yeah, absolutely. So as you mentioned, um, people have traditionally been really aggressive in treating cancer. They just want to throw everything at it, chemotherapy, radiation, and it makes patients quite sick. It also puts pressure on those cancer cells to find genetic ways to overcome these treatments. So what often kills a person is not the cancer. What often happens, I should say, is that the cancer evolves resistance to the therapies, and that is what proves sadly. So the new thinking that I write about in this book is that we could maybe look at tumors instead of uniform and see that they are this universe of mutation. And can we play the cancer cells against one another? Can we pit them against one another so that we're taking medication away in designed intervals, allowing the cancer resistant cells, I'm sorry, the drug sensitive cells to multiply, and then you knock those down at a time where they're most vulnerable. So it's using Darwinian dynamics to make cancer not exactly to treat it differently than we have in the past. I'm sorry if I'm not explaining that great.
I think there's a double hit here. So the chemotherapies, that poisons that have been used for cancer, induce mutations too. They
I'm sorry, that's exactly right. Yes.
produce mutations, and they basically are selecting out for resistance and this Darwinian cell competition. So you have this double hit, and so perhaps, of course, it'd be great if we get to a much more intelligent way to treat cancer, as we're starting to see. But also the idea that, not killing every cell and basically promoting this resistance may not be such
Exactly.
a
You
yeah.
said it better than I did. That's 100% what I was trying to say.
I think this is something really important for people to get because this is already, I mean, we haven't had alternatives until recent years, and we're starting to see more and more. And you made a great parallel with the pesticide and moths story to help people understand that in the book. And so after cancer, the next area I wanted to get into, which is a lot of surprises here for folks, is the immune system.
Right.
Immune system, um, and maybe you can tell us about so these B cells are antibody producing cells,
Right.
somatic hypermutating. They make 10 to the 18th, a quintillion or a
Yes.
million trillion.
They can make it. They don't do it necessarily, but they have the possibility to, yes.
Right. More antibody shapes than you'd ever potentially conceive. So this is a powerful system to help in the way of somatic mutation. So maybe you can help, understand us understand this and the fact that, for example, COVID boosters led to these 400 some unique antibodies that protected against Omicron even before Omicron was
Right.
created.
Right.
So
Yeah.
people should understand that our body is pretty darn smart about this, making this reservoir of diverse antibodies.
Yeah. And this goes back to this big idea that I was trying to communicate, that our DNA changes every single day of our lives. It changes as you and I are talking. We've got mutation going on in our bodies, and there's the downsides, like we were talking about, cells can mutate and become cancerous. But what I have learned in the process was that we have to mutate our cells must mutate in order for us to be healthy, in order for us to fight the microbes and viruses that we encounter. Every time you and I get sick, we've got these B cells that are essentially doing like a Vegas lottery inside their DNA and just reshuffling, kind of like that lever machine, the slot machine you pull in. It just kind of spins things and it shows you a new mixture. And it's doing that because it has to make the right antibody shape that will bind that virus and neutralize it, like mark it as trash, help get rid of it. So we're kind of playing the lottery in a way, and we have these really smart cells that when they do find something that works, they have these feedback mechanisms to kind of make more of themselves. And as you mentioned, it happens when we get sick. We've got this mutation happening. It also happens when we get a vaccine. And oftentimes people are like, wait a second, isn't this what the anti-vax contingency has been saying, that, oh, I don't want to get that vaccine because it's going to mutate my DNA. And what I'm trying to say is, yeah, it's going to mutate your DNA and you want it to. Because there are people that have conditions where their immune cells cannot mutate. They have hyper IgM syndrome. I'm speaking with patients that have this, and it's an extremely precarious situation to be in because your body cannot develop the right defenses, and it can be fatal often. So these patients have to undergo lots of treatments. Some of them do a whole bone marrow transplants in order to reset their immune system so that their immune cells can mutate. But at the end of the day, yeah, a vaccine, a booster shot will just make you have better antibodies through the process of mutation.
Yeah, I mean, this is a critical point because, as you point out, the hyper IgM, the IgM syndrome is as an example. If we couldn't do this, we would die of a common cold. I mean,
Right.
this is
And there are families that have many, past relatives that have died of this before some of the treatments were available.
you really highlight. You're not, of course, we were talking about the hyper IgM syndrome, but you highlighted the AID, this activation induced cytidine.
Yes.
It's a mouthful, but
Yes,
this
it
is
is.
an enzyme in the body, right?
It is, and I'll tell you, I wanted to write more about it, but it gets pretty technical pretty quickly. But essentially, there's all these sorts of mechanisms for our genes to resort, reshuffle, and there might be some ways in which targeting this can push our health in one direction or another. So I think AID is an important example. But generally speaking, just understanding the levers of mutation, understanding that different people's DNA mutates at different rates is also important. So there's a lot
Yeah.
to be learned in this area.
And a couple of things before we leave the immune system. One
Right.
is that, as you highlighted in the book, and also in a paper that was just in the last week, is
Yes.
that these immune cells, can acquire somatic mutations that are the root cause of autoimmune diseases.
Yeah.
So
Yeah.
that
So
we haven't really conceived that as why we get autoimmune diseases, but that certainly seems to be one of the pathways that can occur, right?
I think this is why it's so powerful to appreciate acquired mutation is important for our health. Because as you mentioned, there's just this past week, a new paper that's pinpointing the somatic mutations that are causing immune cells, instead of becoming better at making antibodies, these immune cells have mutated the wrong way to go after the person's own tissues. So if we can now figure out these mutations, now that we understand that they're happening, we can target them. We can work towards new treatments. And to me, that's super powerful.
Yeah. So now we're going to move on to the brain. And this is some great work that's been done. Um, that it was described actually by Ed Young, another great science journalist like you. Each neuron really is a beautiful and unique snowflake. And we have neurons that have 1,500 unique mutations in them. And of course, today was published a paper is that mutations in the microglia in the brain, the immune cells in the brain, have the same mutations as seen in cancer cells.
Yes.
And when they're put into induced pluripotent stem cells in culture, they actually incite profound inflammation. And they are thought in Alzheimer's patients, they're enriched and thought that they're actually these somatic mutations in the brain, in the microglia, are a potential root cause driver of Alzheimer's disease. So here again, the brain. Further thoughts about this acquired mutations in the brain?
Yeah, and it reminds me. So I did want to mention. I was rereading last night your post about CHIP, so clonal hematopoiesis of indeterminate potential. Although I think you and I both agree that maybe the indeterminate potential is not a name we'll stick around with forever. But yeah, so firstly, just the idea that my brain, your brain, our brains have these islands of cells that are kind of genetically distinct blows my mind, no pun intended. And the fact that it has been linked very firmly, that more and more cases of autism are being linked to acquired mutations early in embryonic development. So it's not just Alzheimer's, right? Like this is being looked at as schizophrenia. It's been shown in epilepsy, and actually there's drugs that have been tested based on the insights in somatic mutations in epilepsy. But the idea that the same thing that might be happening in our blood that can cause cardiovascular disease and inflammation might also happen in our brains and cause Alzheimer's. I think we're just entering this whole new era of understanding genetics in medicine, and I think it's exciting. I think that going back to kind of what we were alluding to earlier, people want to know the causes of why they get sick.
Yeah.
And if we can now show them that this might be the reason, I think that might help people.
No, absolutely. We're going to get into how these mutations can keep you healthy or go from bad to good in a
Right.
moment. So during the lifespan, there are some critical things. We talked about that young girl who had the long QT syndrome, acquired mutation.
Right.
There's a classic that I think a lot of people aren't aware of that was one of the great insights early about this somatic mutations, the lines of Blaschko.
Oh, yes. Yeah, really striking, visually striking.
Tell us a little bit about that, is because it happens, in the early stage of life, and it's what all these different skin colors.
Yeah, I mean, it's almost like a kind of marbled look on the skin of these deeply pigmented areas and light areas. And it can also follow the midline. So you might have this marbled look of red and not red skin on one side and not the other. But it's the idea that you can actually see the trajectory of mutations and where they land on the body, excuse me, by, just gonna have a sip of water, by on the surface of the skin. And it was interesting to me that because this was discovered kind of multiple times, and it's such a visual example. I think at the time DNA had not yet been discovered. That happened in the 50s. And so we now have the answers to how these striking things happen. And as I think you and I might talk about, there's a lot that we can understand just by looking at the skin in terms of how mutation can cause something visible.
I think it's because a lot of people are not so up to the importance of somatic mutations. The skin, that example,
Yes.
is one that you can see. The skin.
Yeah, and the lines. I wish I had a picture. Just I have a picture somewhere on my computer, but it's following these pattern lines, and it's kind of the same on each person in a way. Like it's predictable, and I think this goes to some of the research that's been doing, like the Human Cell Atlas. Like all these ideas of following development, following cells and where they are in the body is important.
Yeah, well, when I post this, our conversation, I'll put a picture of
Oh,
that
great. I
along
can
with
send you some. I have many.
we're discussing, and of course, also with the excerpt you already have in the Atlantic, and a brilliant review by my friend Jerry Grootman in the New Yorker. Also, one that was just appeared yesterday in the Wall Street Journal of the book. So now, before I switch to all the beneficial things
Mhm.
of the mutation,
The happy stuff.
the revert and autocorrection,
Yeah, absolutely. Yeah.
I do want to just highlight a few. You mentioned one, CHIP.
Yes.
As we get older, age 70
I
plus.
know you tested yourself, right?
Yeah, I mean, fortunately didn't have,
Yeah.
clones that were of concern. But what I can't get over is here we have valuable information where you have a clone of your blood stem cells, or
Mhm.
in men, the loss of the Y chromosome, which occurs 20+%, 40% of men as over age 70, and they have correlations with diseases. In the case
Mhm.
of the clonal hematopoiesis CHIP, with cardiovascular disease and
Yeah.
cancer and the loss of Y, also with heart failure, Alzheimer's. But we don't test these things, and these are somatic mutations, right?
Right. And the test I think you took was like $950. So it was, even when it's available, it's not available per se.
Oh, it's crazy, and of
Yeah.
course, I would never have paid for it, but they contacted me because I've been writing about CHIP and why
Yeah.
aren't we studying this? You can get it from sequencing, just a
Right.
regular genome sequence, but
Sure.
the deeper you sequence, the more likely the accuracy, I mean, is raised. But these are some examples of somatic mutations in the clinic that we know are important that we
Right.
don't assess. I guess
Yeah.
because it's not
I
practical.
I did want to ask you a question here, Eric, because I read your post again last night, and it mentioned you weren't at the 2% threshold, right? So it's when people reach 70, 80 years old, it's 10 to 20% of people have this kind of threshold level of these mutant blood cells that have mutations in certain genes that are of interest, and this is linked to cardiovascular disease. But if I read your post correctly, it said it was like a 0.3% prevalence of these cells. Is that accurate, or is that just like an artifact?
Of the TET2A
Yeah.
yes, yes.
So does
But
that mean that they've tested and they found some, but just not threshold level?
right, they found various TET2A,
Yeah.
but very low levels cumulatively at point 0.2, I think, or point
I
0.3.
think I'm bringing this up because to me it's you know me well enough to know I kind of lean into the unknown and I lean into the complexity because there is this question of what is the threshold, right? Like,
Yeah.
and there is, and I was telling my babysitter about this. She's quite young; she's in her 20s, and she's like, "Well, I'm young; I don't have to think about this." And then I thought about myself in my 40s, and I'm like, "Well, these cells start cropping up. They don't reach threshold very commonly until you're older. But what does it mean that we have? Is there something that's keeping them at low levels, and what causes them to um, proliferate, to multiply?" And I don't mean to hijack your question and go in a different direction, but it just got me thinking because you wrote about it that there is this question still.
I think that's one of the most important concepts for people to take here to get is that our cells are constantly dynamic. We're going to talk about how they can revert,
Yeah,
autocorrect,
yeah.
natural gene therapy, if you will.
Natural gene therapy. Yeah.
Yeah, actually, why don't we get into that now? Because this is one of the most extraordinary parts of your book, is that you give us many examples of how we undergo correction, autocorrection. And it's something I think a lot of people don't appreciate. And it's also a lesson for
Mhm.
how we could fix our genome, which is obviously the genome you are conceived with is very different than the genome you die with, or the
Right.
genome yesterday is different than the genome today and the one tomorrow. But if we could actually pick up these mutations that we can naturally because of this continued cellular competition,
Mhm.
you give some examples. Maybe you can just take us through one or two of these.
Yeah, sure. So folks listening might know about there's children that are born with severe immunodeficiencies, and so for example, there's ADA SCID, and this used to be called bubble boy syndrome, but I don't really like to use that term. I think it's very othering, and I just don't like it. But people might be familiar with it. And in the 90s, they found that there were a couple of cases. There were these two boys in New York. One of them actually had an older brother that had died of this, and they found that for different reasons, like religious reasons and other reasons, these boys didn't receive some of the treatments that were available. Not that they were like curative treatments. Um, but what they saw was that they were kind of living well and long and had good immune systems. And they were like, what's going on here? These kids should be sick. Why are they not sick? And when they examined their immune cells, they found that they had been born with this ADA SCID disorder, but some of their cells had kind of gotten a secondary mutation or reverted back and basically become functional again. So they'd healed themselves. They were doing great. And there's other examples of this, like we were talking about skin. There are some really interesting images, and I can share those with you too if you'd like, of patients where they have epidermolysis bullosa. It's like this disorder where it's inherited again, where um, what you and I will just be like a little bit of a scratch, perhaps, will turn into a potentially fatal infection. And what they saw was that, and you can see because a lot of the skin is blistered and irritated, but they noticed that patients often have these patches that are completely healthy, and in those patches, the cells have reverted or have self-corrected. And the interesting thing is, what can we learn from those mutations that can apply to patients that haven't spontaneously developed these mutations? Like, well, how can we develop drugs based on these insights? I'm sure you're aware, like with Mendelian disorders, we're kind of always looking at like what inherited mutations can be bad or cause us to live longer, and then we develop some treatments like PCSK9, cholesterol stuff that come from that. So now the idea is, well, what if we can use the body as a landscape to pick out cells? And do
Yeah, that gets us to the fact one of the really important um, ideas you came up with, and now it just appeared in a Nature perspective on cancer evolution, is that we need promolytics. That is, we need ways to pick up these promoter mutations that cause cancer and get to that stage before there's a cancer and try to do this, essentially this natural gene therapy, but now with an actual intervention. So the conclusion of that paper, which I'll link to, is just as you're saying, is when we find these mutations in people that are potentially really dangerous for cancer or other diseases, there's a route here that we should be. We're getting smarter, just like our body can be very smart too.
Well, I think, but you're kind of also alluding to something important, which is sometimes it's not the mutations itself that we're seeing that we should pay attention to, that there are things upstream. So like with the loss of Y, the fact that a large proportion of men as they get older just lose these Y chromosomes, and that it has been linked to cancer and things like that. But the idea is that maybe it's not actually the loss of Y causing the cancer, because similar things have been seen in women, right, where there's no Y chromosome. So maybe we could use AI or technology such as this to sort through the noise, but we kind of got to look further up to figure out what's causing all this happening.
Yeah, I mean, I think the acquired somatic mutation story is accounting for far more than we had given credit to. Another example you bring out in the book in midlife is endometriosis, common condition in women.
Very
that
common.
could be a somatic mutation story, right?
Yes, and again, this is one of those things where I wrote the book, and then the papers just keep coming. So, like, endometriosis is a very, I guess, mysterious illness because it's not cancerous, and yet there's these growths that can happen not just around the reproductive tracts, but they can happen. Like, I know I have friends with endometriosis, and they've got growths happening all over the body, and it's horrible. Now we have the idea that maybe these are somatic mutations that have caused these cells to go rogue. And I think that's really important to understand, because if we then, again, can understand a mysterious illness through the lens of acquired mutation, it just opens up a whole new avenue of treatment,
Yeah, well,
potentially.
it's getting ahead of things. You touched on the Red Queen. Uh,
Mhm.
Maybe you can elaborate on that, because in a way it ties into all this.
In terms of, I mean, the Red Queen being that you have to run twice as fast to stay in place, kind of a
Yeah,
riff on a Lewis Carroll thing, and
yeah,
the idea that with cancer, for example, you kind of got to like beat cancer at its mutational game before it beats the treatments. In
that,
terms of endometriosis, I don't know if, I mean, I think like you say, we're really, really at the early stages. So I don't know how the Red Queen would necessarily apply to that particular one, but I think it's very important in cancer. And even with something like CHIP with the mutant blood cells, we are we're now seeing the initiation of some therapeutic approaches. And I guess your question's making me think, okay, well, if those work, are the cells going to mutate around those? Like, it is a Red Queen type of thing, like
yeah,
you say.
I'm thinking of it especially for cancer,
Yeah.
but getting ahead, and that's we're not doing that, and this could be a really important ticket to prevention. Now although we said we come back to it, I do want to talk a little bit about the environment, because we know how important it is for inducing these mutations, whether it's air pollution,
Yeah.
micro nanoplastics, forever chemicals,
Yes.
uh, UV light. What's your sense about the impact of acquired mutations from all these environmental triggers?
Can I mention behavioral things too? Does that count as environmental, or is that,
Sure,
no?
please.
Because I know that you wrote in your post about CHIP about smoking, right? You know, how smoking can actually promote or maybe it's cessation of smoking, but basically your smoking behavior can influence the prevalence of these mutant blood cells that are linked to heart disease and stroke and cancer. But I think what I found is that sleep has also been linked. So if not that I'm practicing this myself, I should take my own advice. Hopefully now that the book's out today, I can um, follow my advice. But sleep has been linked to fewer mutant blood cells. So I think that we can kind of now, it's exciting. We're almost connecting the dots through the lens of somatic mutation, through the lens of acquired mutation. We're able to kind of see the data that supports these behaviors. And on the flip side, like the environmental exposures or toxins and things like that. What I thought was really, and this is kind of new science that just came in, and as I was finishing the book, but they can now look, because there's a lot of noise, right? If there's a lot of mutation in your body, there's a ton of noise happening. But they can now kind of, through technological computing methods, sort through that noise and actually look at your mutations and tell you if you are smoking tobacco or chewing tobacco, because those have different
Mm,
mutational profiles. And I'm like, that it just blows my mind. But it also kind of maybe will hopefully persuade people to not do these behaviors if they can see that it's not just a theoretical thing that it will mutate your DNA. We can see it.
well, on that note, today published also in Nature Medicine, the journal
Mhm.
that
I was at for 12 years. Yeah.
you from Nature Medicine, many
Yes. That's how I met you.
pesticides were
Mhm.
linked to early onset colorectal cancer, in young
Interesting.
people with an epigenetic signature. So, the fact that there's so many environmental, and we've seen this early cancer, not just colon, but, breast and other types of cancer, that we're getting these acquired mutations unfortunately, from our environmental exposures that have just, the people think that the plastics, induce inflammation, but they
Right.
can induce mutations, right?
And you said something very interesting here because you mentioned epigenetics.
Yeah.
And oftentimes people ask me like, oh, so your book's about epigenetics, because that's kind of what people think about when they think about DNA that changes. And epigenetics is, of course, the markers that sit upon the DNA or the chromatin that the DNA is bound around that silence genes or help flag to activate them. I will say that, so my book mentions it in passing. Epigenetics is super important, but it's really about the sequence changes, so the actual DNA changes. But what's so curious to me is that there might be a dialogue, like a crosstalk between the somatic, the DNA changes in the actual sequence and the epigenetic changes on top of it. And I've just been talking to a bunch of scientists about how, what directionality does this relationship have? Is it what's driving the other?
I think this relationship of environmental exposures, behavioral things, I mean, is fascinating. So
I'll add, Eric, one thing
yeah,
that with the brain science, they found that the somatic mutations were most common in the genes that were turned on most frequently. So I think it's not just about necessarily. I agree with you. I think that definitely toxins and behaviors have a monumental impact. But I think also we kind of have to understand that just the wear and tear of the body, the functions that are just happening to get things going. Sometimes, like just like a light switch in this room, like I turn it on and off. At some point, especially the way things are built these days, that light switch might malfunction just because it's been used so much. So
yeah.
is something happening like that with our DNA?
Well, yeah. I mean, as you point out in the book, by the time a blood cell has reached 100 years, it's got 4,000 unique mutations that
A
have.
white blood cell, right? A lymphocyte, because the red blood cell won't have any DNA. But that's yeah, 3,000 mutations in a. It's not definitively proven. It's just based on the samples of cells and estimates. But that's a lot of mutation.
No kidding. All right, so we have, I think, here the basis. And by the way, there's many other diseases that you get into in the book,
Yeah.
from VEXAS. I didn't know any.
Vexus. Wow. Yes.
Uh,
Yes.
Paroxysmal nocturnal hemoglobinemia, Duchenne or dystrophy. I could
I'm
go.
laughing not at the disorder, but the fact that you say it so well, because it took me so long to practice for the audiobook. But yes, PNH for sure. Yeah.
Yeah, PNH, that's one of my favorites. I mean, how you get into the pig A mutated gene and all that, the dark black urine. All I mean, the book is rich. It's really rich because it gives you a new appreciation. One of the problems we have, the stumbling blocks, Roxanne, maybe you could comment on. Although it's easy to get a skin biopsy to look at these somatic mutations, unless you're getting actual tissue for a reason, heart, brain, or whatever, we
Yeah.
can't we can't get to this. It's only in a workup, and of
Yeah.
course academic studies. So this is one of the things that's holding us back to get full appreciation, right?
It is. I think that one of the hopes I had is that maybe a family will read this where maybe they have a child that's undiagnosable, or has a disease that's undiagnosable, or there's a person, like you mentioned with Vexus, this is a disease that was just discovered in 2020. So all these men had fevers that they could not explain, runaway inflammation that was fatal in some cases. And I think that my hope is that maybe by reading this, people will be able to advocate for some testing, and perhaps that will precipitate change in the medical system. I don't know. I think part of it comes from us, right?
Yeah, well, your point is so important because we've had a program here at Scripps Research called Idiom for idiopathic unknown.
Mhm.
And
Yeah.
when we hunted down the unknown disease, we often would have to get to the tissue level, not just
Mhm.
the DNA, because that's where we found whether it's the heart or the
Yeah.
brain. That's how we found the root cause of the disease.
Yes, and, like, I'll say that, my I have a friend who joked that the book cover looks like a party invitation, and I thought it was really apt because, first of all, I love to throw parties, I love to, but secondly, I was talking to scientists that were doing the same thing with different diseases in different fields that just didn't know about each other,
Mm.
and so if they didn't know about each other, I don't think that maybe an average family physician or maybe, people that are specialized might not know about this happening. So, I mean, maybe my hope is that doctors will also read this and say, "Oh, like, this gives me a different way of thinking about diagnosis," and so that's a hope I have.
Well, it certainly gave me that. I mean, as I say in the
It's
outset,
really nice of you to say.
this is a mind-bending book. If you really want, and it's not it's an easy read. It's not an opus, and you will learn so much about these mutations, somatic mutations. I certainly learned things I never had ever thought. I mean, like just the book that we talked about, The Struggle of the Parts,
Right.
plus all the autocorrect, the revert mutations. This book is so informative, and you've done just a tremendous job of explaining it to people without a biology, life science background. So I just give you so much credit for it. This is going to be a really landmark contribution. So before we wrap up, anything else that you want to get into that I didn't cover, because I can't do justice in a 40 minutes?
Oh, Eric, first of all, thank you for the kind words. I'm such a fan of your work. So just I'm going to be floating on cloud nine with all the nice things you said. I will say I'll just give one last little story because it's something that my friend said was the most interesting thing to her. She was texting me about it, saying this is really blowing my mind. Just that we haven't really talked about the mutations that happen in our reproductive systems that can be passed on. And of course, these aren't somatic mutations, but I have a whole chapter because I've talked to a lot of patients recently, or a lot of parents recently, that have had kids with diseases that have not been in the family. And in these cases, it's not a somatic mutation that happened in embryonic development, which is only in some tissues. These are in all the cells of the body because they happened in the sperm or the egg. And the story that was shocking to my friend that she texted me about was that scientists in the UK looked at the UK Biobank data, and through looking at that data, they found these children that had, I'm getting goosebumps as I tell the story because it's very strange and unusual. They had mutations that are often seen with a type of chemotherapy called cisplatin, but the kids had never had cancer. So they were like, what is going on here? And what they were basically able to figure out is that the fathers had the chemotherapy, and that their sperm had acquired, their sperm stem cells had acquired those mutations. And then the children then developed the disorder. Men make 2,000 sperm every heartbeat, about 1 to 2,000. So there's a lot of cell turnover. There's a lot of opportunity for mutations to happen. But to me, that was
We're the
interesting.
And much more than women with respect to these types of
I
mutations.
mean, that's another thing that my friend was saying. So, so 80% of these de novo germline mutations, 80% of these new mutations that happen in the sperm or the egg that are passed to the children are from men. And, I'm a woman who had a child later in life in my 40s, and the doctors were very nervous about my DNA. But in reporting this book, I was like, well, wait a second, like only 20% of these mutations, obviously the chromosomal abnormalities often come from women, but yeah,
I mean,
a lot.
that's where I think a lot of people, again, don't realize where this is coming from. As you said, these rapidly turnover of sperm that
Yes.
the culprit. So if anyone has questions before we finish, just put them in the message. There's been some comments. Some people are getting the book. Some people saying, I don't know if I can understand this. You can understand it. That's why you're such a well-known science journalist, because you're such a fine explainer to people. And that's why the Atlantic has you and so many others over the years. But anything else from either the folks listening or from you, Roxanne?
I don't have anything to add except that, I just hope people understand that we are all mutating. And it is a complex science story in some ways, but it's pretty simple also that, you and I were talking, we're mutating, and now we're going to figure out what that means for our health.
Yeah. And Wendy just asked, will this talk be available to share? Absolutely. We'll post it soon with all the links from these papers that we've been discussing, the classic book from 1881 of Struggle of the Parts, which I don't know how that guy could have figured that out, Wilhelm Roux, back then, as to what is going on now. I thought we'll post that. We'll post some pictures of these.
I'm taking notes to send this to you, yes.
And, this is a hot area. And as I mentioned earlier, and I think Roxanne agreed, we wouldn't have known what we know now if it wasn't for the era of single cell sequencing. And this has just enlightened it, it's lit it up. And so that's why the book is so timely, because you synthesize so much. You've added a lot of your own insights around that in an era of life science that didn't even exist, several years ago, and now is just flourishing.
Yeah.
So thank you, Roxanne. I really enjoyed the conversation, really enjoyed the book. And I hope that others who want to learn about the importance, not just in diseases, but also in our health. And I do think what's exciting here is that we're going to see ways to prevent cancer as the promolytics, or the treating the high risk mutation to get it out of the way, rather than waiting for a tumor to develop. I see one more question before we wrap up. The genetic autoimmune disorders, mutation on markers that change the cells. I can't actually, it's a long one, but I can't see it all. Let's see. Oh, here it is. If familiar something disease is genetic related, immune disorders, mutation on markers that change. So I'm not sure what that question is. But that's from Jojo. Okay. Sorry. I don't know what, do you know what the question is, Roxanne? I can't.
Maybe the person was asking about inherited autoimmune, or like the propensity to, because, autoimmunity does run in families.
Sure.
And I think I was mentioning earlier, like one thing we're trying to understand, and there's some signs of, is that certain people might have certain propensities to, like, inherited propensities to mutate in certain ways. And I've written for this in The Atlantic, actually, that there's certain inherited propensities to mutate and actually have adverse vaccine reactions, which is kind of a autoimmunity thing in some ways, or immune system gone wrong. So I guess what I'd just say to that is maybe, autoimmunity is complex, and mutation is part of it.
Yeah. This paper that just came out in the past week about the lymphocytes developing mutations that led to autoimmune disease just goes right along. This is why your book just keeps coming. It's
And
getting
one thing we didn't talk about also is aging, obviously super aging, super agers. And I'm thinking about it because the immune system kind of also has a propensity to be wrong more as we get older or just kind of go a little awry. And there is this whole area of science about whether we could or should try to stop mutations from occurring in the body, and how the bowhead whale, which lives 200 years or more, can give us lessons in terms of DNA repair. So
well,
a
I'm
lot to
good.
talk about there.
This will be the end point. But in superagers, people have wondered how much of that is just luck,
Yeah.
stochastic. And so while we're all getting these acquired mutations, maybe some of the people that live healthy lives, 85 plus, they're just lucky in the types of somatic mutations they get.
Yeah.
Or their ability, as you really hone in on, the cellular competition is highly effective to keep away the non-helpful, unfit, potentially cancer-driving whatever mutation. So it's really intriguing because as you say, the immune system is so paramount here. And maybe they keep their immune system healthy just because of various dynamics occurring at this mutation level. So we'll wrap it here. Thank you so much.
Thank you, Eric.
It's been fun, and I hope those who are at all interested in their body will get Beyond Inheritance because there's a lot here. It's really rich. So thank you for writing it. Thank you for 3 years of research to crystallize.
Thank you for always being fascinating, Eric. I continue to take signals from you in terms of where the future of medicine is. So I appreciate you having me here.
Very kind of you. All right, you take care, and we'll get this posted soon with all the links we've talked about. Thank you.
Thank you.
Thanks for joining.